The first step of the cell cycle is interphase. It splits into 5 phases. There's G1, where the cell grows. Then there's synthesis, where the DNA becomes replicated. Then there's G2, which is a part of mitosis. Sometimes after G1, it will go into G0, meaning it can't divide. An example would be a neuron brain cell. Therefore, it just grows. In the G2 of interphase, 2 centrosomes are found by the duplicated of a single centrosome. The duplicated chromosome of phase S cannot be seen individually because they have not yet been condensed.
Now you're seeing prophase, where the chromatin fibers become more tightly coiled. The nucleoli disappears, and each duplicated chromosome appears as 2 identical sister chromatids. The mitotic spindle begins to form, which is composed of the centrosomes and the microtubules that extend from them. Now the centrosomes move away from each other, propelled partly by the lengthening microtubules between them. Now we are in prometaphase, where the nuclear envelope fragments, and the microtubules extending from each centrosome invades the nuclear area. Now, the centrosomes have become even more condensed, and a kinetochore, which is a specialized protein structure, has now formed at the centromere of each chromatic, which gives it 2 per chromosome. Now, non-kinetochore microtubules interact with those from the opposite pole of the spindle,
The cell is now
which
entering
lengthens
metaphase,
the cell.
where the centrosomes are now at opposite poles of the cell, which leaves them at equidistant between the 2 spindle poles. For each chromosome, the kinetochores of the chromatids are attached to kinetochores microtubules, coming from opposite poles. The cell in anaphase is the shortest phase of mitosis. Anaphase begins when the cell cohesin proteins are cleaved, allowing the chromatids of each pair to part suddenly. The chromosomes begin to move apart to opposite ends of the cell until the 2 ends have equivalent collections of fragments of the parent cell's nuclear envelope and other portions of the endomembrane system. Chromosomes become less condensed, and any remaining spindle microtubules are depolymerized, in which mitosis, the division of one nuclei into two, is now complete. A normal cell